Hi, this is Dr. Allen. We're going to talk about cell anatomy in this lesson. Here are your objectives. This is not going to be a comprehensive overview of cellular anatomy. In fact, you could take whole courses on just that. But we're just going to talk about the very basic stuff necessary to continue on in this course of anatomy and physiology. These organelles are going to be highlighted: the nucleus, the mitochondria, the ribosome, endoplasmic reticulum, that's ER, and then the Golgi apparatus. So when we're looking at a cell, this is kind of an image here. This is from visible body. This is a three-dimensional makeup of the cell. Now, um, with that cell, we have this first thing here is this plasma membrane. We've talked about that a few times already in class. You have the cytoplasm, which is kind of this little substance here inside of the cell that holds, that kind of everything is floating in. You have organelles, which are these various structures here. We'll go over into some of those organelles here over the next few minutes. Cytosol, which is the content of the cell here. And then you have the nucleus, which is kind of hidden behind this structure over here. So simple review here of the plasma membrane. It's a phospholipid bilayer. I've said that about a few times now. Anyway, it has the hydrophilic heads, hydrophobic tails. You have these proteins that are embedded in the membrane that allow passage of material much easier than trying to get through the membrane itself. And if you recall, there's also things like cholesterol, there's glycolipids, glycoproteins, and some other things. Within the cell, we have a nucleus. Now, cells are different in the number of nuclei they have. Some cells will have a single nucleus. Some cells will have multiple nuclei throughout them. And then some cells will have no nuclei at all because they were expelled from the cell in order to function. Now, the nucleus is the site of RNA transcription. That's going to make more sense in a future lesson when we actually talk about that. But it's the generation of that molecule called ribonucleic acid that's going to be useful for the process of protein synthesis. As far as the structure of the nucleus itself, the nuclear envelope, this nuclear membrane here, has these relatively large pores in there that allows for the passage of material such as the RNA that's produced, allows it to exit the nucleus fairly easily. It contains chromosomes. The chromosomes are the structures that hold the DNA. In this particular image here, you can actually see the chromosomes with that characteristic X shape. That's not the normal state of chromosomes in the cell. That's like that when it comes time to actually do cell division. But a lot of people recognize the structure here, so I just kind of left it in this image to make it easier to identify. And then within the nucleus, there's this little smaller structure called the nucleolus. Now, some nuclei will have more than one nucleolus, but in general, what the nucleolus does is it forms another kind of organelle called ribosomes. Ribosomes are composed of primarily RNA, but it has some other structures in along it as well. So the nucleolus, ribosomes are created. Those ribosomes can then enter or exit out through these little pores and then be attached here. So you can kind of see in this structure here, all these little red dots, those are ribosomes. So now as we look at the form of DNA within the nucleus itself, before I had talked about the chromosomes, those X-shaped structures, and I said that, well, most of the time the DNA is not in that form. This is kind of what it looks like normally here inside of the nucleus, right? So this is this thing called chromatin, which is where all that DNA is sitting. So we'll just kind of go over what this looks like in this diagram, right? So first we have this DNA that's double helix. The double helix structure has those nucleotides with those base pairs A, T, C, and G. Well, that DNA strand will wrap around these little structures called histones, and you can create what's called a nucleosome. A nucleosome is 8 of these little histone, these little discs here, 8 of those histone proteins wrapped up twice by a DNA. See how there's a little bit of a wrap there. And that creates this thing called chromatin. Now microscopically, this is what this little image is right here, an actual image of chromatin. So you have your DNA and these little knobby thingies, those are the nucleosomes. Well, that chromatin will also curl up, wrap up in a nice tight coil here. That's called a helical fiber. And that's right here, it's this, right? See? This helical fiber will then kind of like bunch up together and form what's called a chromatid. Now, a chromatid is half of this X-shaped chromosome. A chromosome, this X-shaped thing that you're used to seeing, is basically a duplicate of these two chromatids. So when it comes time to cell division, these two chromatids here will split apart. Each of these chromatids are identical to the other, except in the case of, say, like mutations, but for the most part, in normal circumstances, they are identical. Next up, we have mitochondria. The mitochondria are the powerhouse structures of the cell. I used to learn it as the mighty mitochondria because it generates lots of power, if you will. And that power is in the form of, that's right, ATP, adenosine triphosphate. So how does the cell get ATP? Well, there's a few ways of doing it. The most efficient way is through the mitochondria, through a process of aerobic respiration. You can kind of see here, I have it in 3 different layers. These are different levels of dissection, right? So here's kind of like the whole structure here. This is kind of like this first layer taken off, and then you can kind of see the inside of the mitochondria are these little convoluted walls right here, right? So these all 3 here are mitochondria. I just dissected it out using visible body. Previously I had talked about ribosomes as the ribosomes are created in the nucleolus. They are the site of protein synthesis. In this particular diagram, in this image, the ribosomes are all these little tiny red dots. These little red dots are either floating freely, you can kind of see over here, these red dots are floating freely in the cytoplasm, or it can be attached to this thing here called rough endoplasmic reticulum. Rough endoplasmic reticulum. So ribosomes are either free in the cell or attached to this rough endoplasmic reticulum. Now, the rough endoplasmic reticulum, this diagram here points to this right here. This is what's highlighted in this kind of this off blue color. You can see all the little red dots on that rough ER. We'll sometimes say ER, endoplasmic reticulum. This is continuous with the nuclear membrane. And there's going to be a diagram later kind of showing that a little bit better. And so the reason why it's called rough is because it's studded with these ribosomes. And what do ribosomes do? Well, they're responsible for protein synthesis. So therefore, if I say, what is rough endoplasmic reticulum? What is its primary purpose? Well, it's to create proteins, synthesize proteins. And that's why there's all these ribosomes on here. It's just the way that it's the proteins are generated and distributed throughout the cell is differently if it's done in the rough ER compared to as if it was done out in the cytoplasm. So because there is rough endoplasmic reticulum, there's also smooth endoplasmic reticulum. So the smooth endoplasmic reticulum does not have ribosomes on it. The smooth ER will function to create other substances. For example, steroid hormones is one thing that could be created in the smooth endoplasmic reticulum. But the smooth ER does not generate proteins because there's no ribosomes on it. Next up is the Golgi apparatus, sometimes called the Golgi complex. Um, it's a packaging and distribution center, right? What does it distribute? Essentially proteins that are made in the rough endoplasmic reticulum. So within this membranous structure here, the rough ER, a protein is created, and then it travels over here to the Golgi apparatus, where it'll be processed in complex process. It'll be processed and then sent out of the cell through these little transfer vesicles. You see there's an example right here. You can kind of see them budding off of the Golgi complex right here. What they're going to do is they're going to go out to the cell and then exit out the cell membrane through these transfer vesicles. And that's how proteins made inside of the cell can be distributed outside of the cell. And then finally we have the endomembrane system. This is an image from your text. Just to kind of orient you, we have the nucleus. Remember the nucleolus here. You have this rough endoplasmic reticulum. You see it's like these little folding layers of membranes here as, you know, it's kind of cut off here. And then you have the Golgi apparatus here. Again, these are all endomembranes because they're kind of these membranous structures. And then the plasma membrane here is an integral part of it, even though it's not technically an endomembrane, because what happens is that these little transport vesicles will come out and they'll merge with the plasma membrane, and that's how those little proteins are expelled out of the cell through interaction with the plasma membrane. So the endomembrane system consists of the nuclear envelope, smooth endoplasmic reticulum, rough endoplasmic reticulum, the Golgi apparatus, and then these little transport vesicles. All right, that's it for the basics of cell anatomy. We'll see you on the next video.